Welcome to Biotanime.
Structure of bacteria. Bacteria have characteristic shapes like corky, rods, spirals, etc. and often occur in characteristic aggregates like pairs, chains, tetrads, clusters, etc. Surface structures of bacteria. Flagella. The flagella of motile bacteria. A basal body anchored in the plasma membrane and cell wall gives rise to a cylindrical protein filament. The flagellum moves by whirling about its long axis. Pili or fimbriae. Pili are slender, hair-like, proteinaceous appendages on the surface of many bacteria, particularly gram-negative. They are important in adhesion to host surfaces. Capsules. Some bacteria form a thick outer capsule of high molecular weight, viscous polysaccharide gel. Others have more amorphous slime layers. Capsules confer resistance to phagocytosis. Cell wall. Cell wall is composed largely of peptidoglycan, also known as mucopeptide or murine. In thick cell walls, other cell wall polymers such as the teichoic acids, polysaccharides, and peptidoglycolipids are covalently attached to the peptidoglycan. For gram-negative bacteria, outside the peptidoglycan layer is an outer membrane structure about 7.5 to 10 nanometers thick. In most gram-negative bacteria, this membrane structure is anchored non-covalently to lipoprotein molecules known as bronze lipoprotein, are covalently linked to the peptidoglycan. The lipopolysaccharides of the gram-negative cell envelope form part of the outer leaflet of the outer membrane structure. The peptidoglycan layer consists of a glycan backbone of repeating groups of beta-1, 4 linked disaccharides of beta-1, 4 N-acetylmuramic N-acetylglucosamine. Tetrapeptides of L-alanine, D-isoglutamic acid, L-lysine or diaminopamelic acid. D-alanine are linked through the carboxyl group by amide linkage of muramic acid residues of the glycan chains. The D-alanine residues are directly cross-linked to the epsilon amino group of diaminopamelic acid on a neighboring tetrapeptide, or they are linked by a peptide bridge. Teichoic acids. Wall teichoic acids are found only in certain gram-positive bacteria such as Staphylococci, Streptococci, Lactobacilli, etc. Teichoic acids are polyol phosphate polymers, with either ribitol or glycerol linked by phosphodiester bonds. Substituent groups on the polyol chains can include D-alanine, N-acetylglucosamine, N-acetylgalactosamine, and glucose. These highly negatively charged polymers of the bacterial wall can serve as a cation sequestering mechanism. Lipopolysaccharides. A characteristic feature of gram-negative bacteria is possession of various types of complex macromolecular lipopolysaccharide. Usually, BLPS molecules have 3 regions. The lipid is structure required for insertion in the outer leaflet of the outer membrane bilayer. A covalently attached core composed of 2-keto-3-deoxyoctanoic acid, heptose, ethanolamine, N-acetylglucosamine, glucose, and galactose. And polysaccharide chains linked to the core. The polysaccharide chains constitute the O antigens of the gram-negative bacteria, and the individual monosaccharide constituents confer serologic specificity on these components. Intracellular components. Plasma membranes are referred to variously as cytoplasmic, protoplast, or, in gram-negative organisms, inner membranes. They are composed primarily of proteins and lipids. Protein to lipid ratios of bacterial plasma membranes are approximately 3:1. Multiple functions are performed by the plasma membranes of both gram-positive and gram-negative bacteria. Plasma membranes are the site of active transport. Respiratory chain components. Energy transducing systems. The H+ at base of the proton pump. And membrane stages in the biosynthesis of phospholipids, peptidoglycan, LPS, and capsular polysaccharides. The plasma membrane is also the anchoring site for DNA and provides the cell with a mechanism for separation of sister chromosomes. Other intracellular components. In addition to the nucleoid and cytoplasm, the intracellular compartment of the bacterial cell is densely packed with ribosomes of the 70S type. Other granular inclusions randomly distributed in the cytoplasm of various species include metabolic reserve particles such as poly-beta hydroxybutyrate, polysaccharidae and glycogen-like granules, and polymethyphosphate or metachromatic granules in endospore.
Thank you. Please like, subscribe and share. See you again in a new topic.